Start today with a show of hands. A small percentage of Europeans are HIV resistant. We're going to leave off the rest of that quote for now, just that part. True or false? Okay, anybody think that's false? Does anybody know that's false? It's not about thinking, it's about having evidence. Okay, so a lot of you found a lot of great literature on this topic, and a lot of your comments basically said the same thing, that yes, this part was true, that there are some Europeans that are HIV resistant. And then from there, everything went downhill. Yeah, exactly. By the way, if you want to see what I've been posting on the internet, you can go to that website if you want to, and I'm posting summaries of what we've been deciding when we're fact-checking all of these so-called DNA facts. So this much might be true. But then when we add in the because of genetic mutations caused by the plague of the Middle Ages, then your responses started varying drastically. So almost everybody, I think, wound up saying, no, this fact is not true, not a fact. But there were a number of reasons. I thought I'd share this with you as well. I decided to ask Twitter, although I didn't have that many respond. 10 votes, so if you want to follow me on Twitter, maybe you can help me a little bit with my, I've only got like 2,000 followers or something, so I need more.
Only 2,000?
Maybe it's less than that, maybe it's 900.
Wow,
I don't know.
Still more than me.
Go check me out, I don't know.
God, I have like...
So, everybody, yeah, social media is a thing. Everybody on, or 9 out of 10 people on Twitter say that's also not true. So why is this not true? What were the reasons you came up with? There were a number of very different reasons, all of which were very valid. Yeah.
Because some suggest that the HIV resistance came before the plague or another event.
Right. So one of the things you came up with was right there, the CCR5 delta 32 mutation. We can talk about details if you really want to know what that means, but that mutation existed before the Middle Ages. So some of you found, right, the plague, I think they were talking about the bubonic plague probably, Middle Ages, Europe. And then there was some paper where somebody had dug up some bodies from the Bronze Age and done DNA testing on bone samples from really old people before the Middle Ages and found that this mutation also existed way back then. So this mutation that allows HIV resistance existed before the plague, or at least before a mass outbreak of plague in Europe in the Middle Ages. Other of you came up with a bunch of different studies that talked about the fact that, well, the plague, we don't know exactly what caused the plague, if it was viral or bacterial. Most people think, most scientists think, that it was a bacterial agent that caused mass death in Europe. But we know that this mutation from recent research by virologists, that this mutation only protects cells against viruses getting into the cells. This protein, CCR5, is a protein that's on the outside of immune cells, and viruses bind to that protein to try to get into the immune cells and destroy them, like HIV binds the CCR5 protein. So you could imagine maybe you've got a cell, and it's got a protein on its surface. That's the CCR5 protein. And if you have a virus that has a particular ligand, a protein on its surface that can bind and dock to the CCR5 receptor, that's the way the virus gets into the cell. So this deletion, supposedly, the CCR5, this delta sign, Greek delta means deletion. It's a deletion of part of the protein. So if you get rid of part of this binding receptor, then the virus doesn't have anything to dock to on the cell, so it can't get in the cell. So presumably that's how this mutation facilitates HIV resistance and maybe resistance to a bunch of different viruses getting into cells. And then another set of comments and papers that a bunch of you found had to do with the fact that there was another study where they also dug up some bodies and genetically tested them. They had some bodies that were from the Middle Ages in regions that did experience the plague, and age-matched corpses, people that died around the same time but were not in areas of the world where there was plague, and found that there was no difference between how many people had this mutation in places that did have the plague and lots of people died, or didn't have the plague and nobody died of the plague. So you would have predicted, and these scientists predicted that if CCR5, if this mutation helped protect you against the plague, it would be more common in places that had been exposed to plague. So that was another way that people argued against this fact. So and this is not surprisingly, it has to do with HIV and AIDS, and it has to do with genetics. There's a ton of research on this, and you found a load of great studies. And then most critically, in my mind, so if you focus on any of that, this is the most important argument against whether or not this fact is true. It's semantics, really. This is why we're talking about this fact in this topic, mutations. What causes mutations? What have we talked about so far in class that's a cause of mutations?
UV rays.
UV, ultraviolet light, making thymine dimers, cross-linking nucleotides together, and then DNA polymerase doesn't know how to replicate the DNA, so it sticks in two random nucleotides, causes mutations. What other things have we talked about? So carcinogens, including benzopyrenes, these molecules that are found in charred meats, tobacco smoke, and other things, right? They covalently attach to our DNA and cause similar problems to UV thymine dimers, that replication can't happen across one of these benzopyrene molecules when it's stuck onto a nucleotide. Sometimes DNA polymerase makes random mutations by itself. It's not perfectly accurate. Even the enzyme that copies our chromosomes isn't always perfectly accurate. Sometimes it accidentally puts the wrong nucleotide in. What does not cause mutations are diseases, at least most diseases. The cold, the flu, viruses and bacteria don't cause mutations. So what do you think this quote really meant? This is going to be this is an evolutionary perspective. So once you get to Biology 105, this will make maybe a lot more sense. Can I have some creative artistic license for a second? I'm not such a great artist, so let me draw you 10 humans. I could put heads on them too, but that wouldn't really help. So let's just stick with, let's see, 6. I'm also not good at counting. Is that 10? Let's say that one of these individuals, this one, had the CCR5 delta 32 mutation. Right, so these are just people happily wandering around Central Europe in the Middle Ages, and then the plague comes through. Who is the plague going to kill most or fastest or most efficiently? Of those 10 blobs, which blob gets killed last or least easily? The one with the mutation. So what the mutation does, it is not caused by a disease. But if everybody that doesn't have, or if most of the people that don't have that mutation die, we started with 1 person in 10 having this mutation. But after the plague sweeps through, now how common is this mutation? It's 1 person in 3. So it goes from 10% frequency, it's a rare mutation, to 33%. It's pretty common in the population because at that point in time it confers an advantage. So it's not so much that the plague caused the mutation was already there, as or those of you that at least looked up this paper, the Bronze Age dead people, dig them out of the ground, some people already had this mutation. It just changed in frequency. It became more common in the population because when the plague or whatever caused selection came through, it was a useful mutation. And then, of course, there was the debate again of whether or not this was caused by the plague or viruses. Some of the papers you found suggested there was actually HIV that was a component of or the main cause of the plague. We don't really know what caused the plague. It was just a mass death in multiple parts of the world. So it could have been HIV, as far as I read. But then I didn't read too deeply, so I might be wrong. So a number of you said, well, this is not true just because plague doesn't cause mutations. Thymine dimers cause mutations, DNA polymerase errors cause mutations. It's the exposure to the plague that kills people that don't have this particular mutation. Any questions on that concept? So presumably, this is another example of a mutation that's neutral. That is, it doesn't matter if you have it or not, normally, but under certain circumstances, like if the plague comes through, then it becomes beneficial just because your environment has changed. Kind of like sickle cell anemia. It's bad if you've got the two copies. If you're homozygous, if you have both mutant copies, you get sickle cell anemia. If you're in an area of the world where malaria is prevalent, it's good to have one of your two copies be mutated because that prevents you, or at least reduces your probability of getting malaria. So it's dependent on your environment, not just on your genotype. It's both. So no more of this, other than to remind you that we talked about probability distributions, and most geneticists would say that the null or expected distribution of mutations would be every single nucleotide across a gene, because mutations happen, we think, happen randomly. They're just as likely to happen at any spot on the chromosome to any other spot. That there probably isn't going to be much of a difference in where mutations land on the chromosome. Yeah.
Should anything like shift that graph to make the percentage of it think there be mutation higher or cause a spike?
Sure. And that's what we're going to see in a minute, but let's take a minute to think about this question. What sorts of things might you expect to cause a difference in this sort of spectrum, or the frequency distribution? How likely it is that you get a mutation? Are there any processes of mutations that we've talked about yet that are non-randomly distributed, or you would expect not to be random? That is, target specific nucleotides, for example. Yeah.
So usually, you know where it starts. It usually starts at a large spot of A's and T's. It might be somewhere where there's another A's and T's spot.
Okay. So what, along the same lines then, what mutation process have we looked at that's specific to T's?
Thymine dimers.
Right, so ultraviolet light specifically reacts with two T's that are next to each other on the chromosome. Where are we likely to see two T's next to each other on the chromosome? Are those necessarily distributed at random across the chromosome, or might there be areas that have more pairs of T's than others?
Aren't they at the ends?
Ah, interesting.
But they're in the middle.
So yeah, where do you find pairs of T's? Turns out, hadn't thought of that, but our telomeres, the ends of the chromosomes, we'll get to this eventually, but it turns out the telomere sequences, it's another reason to do microsatellite notation, in humans are TTAGGG. The ends of all of our chromosomes are capped, the telomere sequences of the chromosomes are TTAGGG repeated hundreds of times on both ends of every chromosome. That's how our chromosomes are capped. So yeah, there are plenty of TTs there. So you might imagine that thymine dimers would preferentially happen at telomeres. So you might see something that looks like that, where the probability, if you're standing in a field and you're white and you get attacked by ultraviolet light, then your probability of having thymine dimers might be increased towards the telomeres because there are lots of TTs there that could be attacked, so to speak, by ultraviolet light. More generally, we talked about isochores and the fact that the distributions of A's and T's and G's and C's are not necessarily random. There are some regions of chromosomes that have more A's and T's than others. So those might be regions also that would be more likely to have ultraviolet light damage. What we're about to see is a really extreme example. And I don't know if this example is common or rare, but it's really striking. We talked about BRCA1 already, the gene that does DNA repair. So it's good to have it. It's bad when it's mutated because it doesn't operate anymore. There's another protein called P53, which is like BRCA1. It's another DNA repair protein. It's good to have it. It's basically an anti-cancer gene. Its normal role is, when it's mutated, then that is often associated with the development of cancer. So depending on the type of cancer you're talking about, most people that have certain types of cancers will have mutations in this specific gene, P53. So like BRCA1 is associated with breast cancer, P53 mutations are associated with other types of cancers. It's good to have the protein, bad if it's mutated. So if these genes become mutated, I'll answer my own question on the slide as bad. More likely develop cancer when you have mutant versions of those two proteins, either one by themselves. So in this case, P53 tends to be mutated in a lot of lung cancers. Something like, at least at the time of this publication we're going to look at, 60% of lung cancer patients in a sample of lung cancer patients that were in a particular database that the scientists had access to, 63% of those had mutations. 60% of lung cancers had P53 mutations. And most of those were of a particular type of mutation, a single nucleotide polymorphism, but even more specifically than that, they were all situations where a G that was initially in P53 had mutated and been replaced with a T. G to T point mutations. P53 normally has a G at one spot in the gene, became a T in a lot of people that had lung cancer. Which makes you start wondering, I hope, I wonder if this specific mutation is causative of a particular type of cancer. And that's what cancer biologists and geneticists were interested in, is how does this mutation happen? Why does it happen so frequently? And maybe to a certain extent, why does it happen a lot in lung cancers? So then they have to do something to test, or at least to study what's going on. So the hypothesis is that P53 is involved in lung cancer. So P53, lung cancer. What molecule have we been talking about that might be related to mutations in cells that are in the lungs?
Oxygen?
There's a particular, it's the one we already mentioned at the start of class. We've got thymine dimers with ultraviolet light. It's the hard one to produce because it's, or pronounce because it's an actual chemical name. It's the one that's in cigarette smoke. So it's a carcinogen, which all carcinogen means is a chemical that is likely to cause cancer. And they often do so by making DNA mutations. These are the benzopyrenes. Cigarette smoke, charred grilled meats, probably at very low concentrations, so it's probably not hazardous for you to go have a grilled hamburger or Impossible Burger or whatever. So they hypothesized that exposure to benzopyrenes specifically causes mutations in P53, and that causes lung cancer. So let's see, if I draw that in order, that starts with benzopyrene, and then they hypothesized that benzopyrene mutates P53 in particular ways, and that causes lung cancer. If we did it in a flowchart sort of way, that would be the order of events. Exposure to benzopyrene, mutation of P53, development of lung cancer. So how would you, if you were a scientist, you are a scientist, if you're an epidemiologist or a cancer geneticist, what sort of an experiment would you do to test this? Bunch of monkeys smoking in a room. So we don't usually do these sorts of studies on humans, but this is a human disease, cancer. So would you study this process in yeast? You could, but what would be a drawback of studying this in yeast? They don't have lungs, fair enough. So it might be good to study a model that has some lung cell type properties. So you could do this in mice. You could expose mice to benzopyrenes. That would be a way to go. It's a little bit expensive because it costs a lot of money to house a lot of mice and take care of mice. And it takes years to get university approval to study vertebrates. Better than studying humans, but still.
And cigarettes are expensive.
And well, I don't know. They might have a different source of benzopyrene. But yeah, you can imagine getting hired to be the person who smokes the cigarettes and then blows into the mouse cages. Maybe an automatic vaping machine. Just blowing smoke. So what they decided to do, good ideas, what they decided to do was to do this using tissue culture. In other words, you basically get a petri dish and you fill it with cells. So cells grow on the plate of this petri dish, basically. And they chose to work with appropriate cell types. So these scientists grow human epithelial cells, bronchial epithelial cells, so in the air, cells from the airway. So presumably some types of cells that would be exposed to environmental pollutants, like smoke. So you have bronchial epithelial cells, and then we just have to blow smoke on them and find out whether or not, then we take the cells, look at the chromosomes, find out at the p53 gene were there mutations. Well, instead of blowing smoke on them, they actually just used raw, pure benzopyrene in small amounts, but they can directly apply chemicals to these cells. They don't have to do anything about smoke or vape machines and so forth. So then they do, after they get the chromosomes out of the cells, they can use particular techniques that we don't need to know the details about to figure out where benzopyrene attacked the DNA molecules in the cells. Remember, benzopyrene itself doesn't mutate nucleotides. It doesn't change an A to a G or a T to a C. It just attaches itself covalently to the nucleotide, which causes mutation in subsequent chromosome replication processes, DNA replication. So they weren't looking specifically for mutations. They were actually just looking, if you looked across a chromosome, if you looked at every nucleotide on the chromosome, they're basically asking where is benzopyrene, BP? Where is it physically attached to the chromosome? They're not looking at DNA sequence, in other words, they're looking at chemical structure. They're trying to figure out where on the chromosome are these chemical molecules attached to chromosomes in these cells that they've treated. So this gets back to the graph that we've been talking about for 3 days. Predict where mutations are going to occur on a chromosome. Is it going to be a random distribution of benzopyrene stuck all along randomly to chromosomes, or are we going to see something else? So if you are already looking at the slides, you see that they saw something else. Not a random distribution of attachment points of benzopyrene DNA. So when they look specifically now, not just at the whole chromosome, but specifically in the p53 gene, this is what they see. So help me analyze the graph, please. What's on the x-axis? What's across the bottom? Codon numbers. We haven't talked about translation yet and protein sequence and so forth, but that's gene, left to right, nucleotide position, in other words. And then what's the y-axis? Number of mutations. Wait, but they were looking at places where benzopyrene attaches to DNA. They weren't looking at mutations. So there are two types of data in here. The y-axis absolutely is the number of mutations, but these are not data that these scientists collected themselves. These are known, like that I mentioned briefly. We've got a database of cancer patients, and they already knew that p53 mutations occurred in a lot of these cancer patients with lung cancer. So they looked in the database of lung cancer patients, and so they could count where in p53, along the x-axis, where in p53 do most cancer patients have mutations in this gene? So in general, lung cancer patients have a lot of mutations in this region of this gene, from codon 120 to codon 300. Right? There's some people that have mutations, a few, that have mutations at pretty much every spot along the gene. But there are peaks. What do the peaks mean?
That's where they're more likely to happen.
Well, yeah, so is it where they're more likely to happen? This kind of gets back to the, does the plague cause mutations argument. What group of patients are we looking at? These data come from a specific type of person. Who are these from? Lung cancer patients. So if we had another graph of non-lung cancer patients, this graph would probably look totally different. So in particular, what this suggests then is that mutations in people that get lung cancer are not randomly distributed. You're not just as like, if you're a lung cancer patient and you look at this one gene, you're not likely to have a very small chance of mutation at every position. Mutations at specific parts of the gene are likely to make you a lung cancer patient and be part of this data. In other words, the peaks, codon 273, codon 248, codon 157, those peaks represent spots in this gene that are almost always mutated in people that have lung cancer. That doesn't mean that these mutations cause lung cancer necessarily, but it's a pretty strong correlation. In other words, if you could sequence your gene, if you could sequence p53 in you, and you saw that you had one of those 3 mutations, that means you're probably pretty likely to develop lung cancer because those are very frequently found in other people that already have lung cancer. So that's one of the 2 types of data that are in this table. Frequency of mutations in p53 as they relate to people that have lung cancer. At the bottom, underneath the x-axis, there are 3 little parts of the gene that are called out specifically down here, and there are asterisks. The asterisks are what these scientists were measuring, which was where benzopyrene specifically attaches to nucleotides in this gene when cells are exposed to that chemical. And isn't it fascinating? They all correlate the positions of benzopyrene and where it attacks or chemically reacts with DNA in this gene happens to correlate pretty precisely with where people that get lung cancer have mutations in their p53 gene. More than that, which nucleotides in all 3 of those cases are being bound by benzopyrene? All 3 of them are Gs. And I recall, I think a few slides back, I mentioned that one of the things that the researchers already knew before they started this experiment was it was already known that p53 mutations that cause cancer tend to be G to T mutations. So it seems like there's something relatively specific about how benzopyrene reacts with DNA. We don't know what it is, but it looks like there's at least a strong correlation between where this chemical prefers, if you want to call it that, to attack and react with DNA molecules and causes specific types of mutations that can predispose or cause lung cancer. So the authors concluded this manuscript, which was provided as part of the lab manual, or course manual for this topic, mutation, was that they were trying to define a link between cigarette smoke and lung cancer. And so it's kind of a tenuous argument. You can believe it or not. It's just an argument. It's not fact necessarily. That if cigarette smoke contains some benzopyrene, and if your epithelial cells, bronchial epithelial cells, react with benzopyrene in this way, and those mutations cause mutation, or those benzopyrene molecules attach themselves to DNA at spots where we know that there is an association between mutation and lung cancer, you can connect the dots in the chain of logic. It doesn't necessarily prove that benzopyrene causes lung cancer, but there's a pretty strong association. Yeah?
Wasn't there also 40% of lung cancer patients that did not have this association?
And that's where, oh geez, you're going to make me really talk about real genetics. We tend to, for classes, oversimplify things and make rules and say this is how it works. But there are a lot of traits in humans that are not simple. That is, one mutation or one change does not cause a trait. There are some classic cases of that, like sickle cell anemia. But there are lots of traits where it's complex. In other words, multiple mutations maybe have to happen to cause a particular trait. So there may be, and there probably are, multiple different ways to get multiple types of lung cancer. This might be one of them. I could imagine a mutation in BRCA1 maybe predisposes you to lung cancer. It's another gene that's involved in DNA repair. So presumably you want to keep all of your DNA repair genes intact to be the least likely to get any sort of cancer. That's what I would assume. Okay. So that's our relatively brief survey of mutation. For the rest of the semester, though, as I suggested at the start of last class, we're on a trajectory, and everything builds on everything we've learned before. We're going to talk a lot about SNPs, microsatellites, mutations, as we go through the course. That was just a brief introduction to how some types of mutations occur. But mutations are really interesting to geneticists because they create the diversity that we see around us, like the human diversity in this room, and that's what biologists like to study. Often, diversity. How do our cells work? Speaking of which, we're moving on from mutation to mitosis and meiosis. Next topic in the chapter, in the manual. So some of these chromosomes have mutations. We're all mutants, we're all genetically different. Our parents are not genetically identical, so we don't look exactly like our parents. And all of these chromosomes that you see up here, even though these are 2 copies of chromosome 1, for example, in the upper left, they're not genetically identical. So how do we know they're both chromosome 1? There are a few different possible answers. Why do we call those both chromosome 1?
Isochore banding.
So banding, right? Reaction of dyes with isochores give a particular banding pattern. So chromosome 1, we can tell that those are 2 versions of chromosome 1 by banding. But that banding is based on a more basic principle. If the chromosomes band the same, then it means they have similar isochores. That is, the distributions of G's and C's versus A's and T's, frequency as you go across the chromosome. What else does that mean about chromosome 1 and chromosome 1? If you looked at the DNA sequence of the chromosomes, what would you see? Would they be, how similar to each other would they be? Pretty similar? Not so similar? Pretty similar. They're both chromosome 1, which means they have very similar DNA sequences. That is, they have almost the same DNA sequences, except for those mutations that differ between your mom and your dad. They're both chromosome 1 because they have the same genes. There are a number of ways that geneticists, in other words, identify which 2 genes are the same, which 2 chromosomes are the same. Chromosome 1, chromosome 2, chromosome 3. Banding relies on DNA sequence similarity. That's what isochores are. And these 2 chromosomes have the same genes on them in the same order, which is another reason they have the same banding pattern. So there's a particular word that we call 2 copies of the same chromosome in one individual, homologs, or homologous chromosomes. Homo means same. Log means log-shaped chromosome. I don't know. Homologs. The 2 different versions in a diploid, like we are, 2 copies of every chromosome, the chromosomes that are similar in sequence. They're identical in sequence or closely related in sequence. They only differ because of these random mutations. Have the same genes in the same order. That's the definition of a homolog. So how many pairs of homologs are there in humans? We've got 23 pairs. So there are 23 homologous pairs. useful wording we're going to use a lot throughout this semester. Homologs. So the other important thing to understand about homologs is that one of those comes from your dad. I'll usually refer to these, although this is not standard genetic nomenclature necessarily, as P, parental, from dad. And the other version of the chromosome, the other homolog, is maternal. It comes from your mom. So every time you look at a karyogram, you're looking at the 23 chromosomes that you inherited from your dad at fertilization and the 23 chromosomes that you inherited from your mom at fertilization. That is assuming, of course, that everybody is diploid and doesn't have any aneuploidies or trisomies, for example. But normally, one copy of every chromosome. Any questions about that? Okay, so in that thing that I circled in blue, upper left-hand corner for those of you that are blue colorblind, which is super rare, so I don't think anybody has blue color blindness in here. If you do, please let me know afterwards. I'd love to study your genetics. It's super rare. It's often found more in females than males too, which is weird because red-green color blindness is usually a male thing, but females tend to have yellow-blue color blindness. It's really rare, much rarer than red-green. So what I circled in blue up there, how many double helices did I circle? We normally call a double helix that looks like this, with base pairing, double helix. We usually refer to that thing as a chromosome. A single double helix. So how many did I circle?
2.
Okay. Let's take a look. So I heard, does anybody have another answer? 4, 2, 2, 4, 6, 8. Who do we appreciate? Chromosomes. Any answers other than 2 or 4? We're going to take a vote then. How many people think I circled 2 double helices? Raise of hands. Okay. How many people think I circled 4? Fewer, maybe 5 or 6 in the audience. Let's do the same exercise on the other metaphase chromosome spread. There, I've circled, well, I circled part of an extra object. Let's ignore my fat fingers. How's that? Better. How many double helices did I circle on the right side?
2.
2 or 4?
2.
Does anybody disagree? I'm seeing a lot of 2s. Okay, this is the reason that we do this now, before we really start talking about mitosis and meiosis. This will hopefully blow your minds, but only in a good way. The picture on the left side is the same as the picture on the right side. Not from the same cell, but they both represent chromosomes at the same stage of the cell cycle. Because there's only one time during the cell cycle when chromosomes compact in structures that we usually refer to as chromosomes, that we can actually see using a microscope. Does anybody know what stage of the cell cycle?
Metaphase?
So this is, yeah, it's metaphase. This is not. In other words, hard to believe as it is, and I'm going to spend the rest of class-ish trying to be very explicit about what we're looking at here in these pictures, because it's really important. That one little stick, I circled 2 little stick-shaped things on the left side. So I'm going to do are circled. How many double helices? So here on the right, I think we all agreed on how many double helices that was. It's 2. So if I draw that in black, that's 1 double helix and another double helix. Ooh, man, that's horrible. And they're connected together at the centromere. So the arms of the 2 double helices are spread apart. Left side of the screen, what I'm telling you is that this object is a pair of double helices. And so is this one. That's early in metaphase. So over here, there are 4 double helices circled. And here, there are 2 double helices circled. You should definitely ask me some questions at this point, because you should probably still be a little bit confused about what's going on. That would be totally normal. Yeah.
How does the removal of the cohesin make it go from 4 to 2?
Ah, perfect. Did you hear that in the back? How did the removal of cohesin make it go from 4 double helices to 2? Right, this is the mathematical, not trick, but explanation. These are both metaphase chromosome spreads. So here I've circled 2 homologs, the one from the copies of the chromosome from dad and the copies of chromosome from mom. Here, we're looking at one homolog. That's either the one from dad or the one from mom. So the question is, where's the other homolog in this metaphase spread? There's another copy of chromosome 1 here somewhere. Where is it? Like this one? Something that's a similar shape and size. One, probably that one or that one. So let's say that's the other copy of chromosome 1. Now we have 1, 2, 3, 4 double helices on the right. Those are the 2 copies of chromosome 1 from your dad and the 2 copies from your mom. Remember, this is after DNA synthesis. So we've got the original version of dad's chromosome 1, the newly synthesized version of dad's chromosome 1, the original version of mom's, say, chromosome 1, and the newly synthesized version of mom's chromosome 1. That's why there are 4 double helices of chromosome 1. Here, they're just separated a little bit because it's later in the cell cycle. This is still metaphase, it's just a little bit earlier, before the cohesin has been removed. So those 2 double helices from dad are stuck all along their length, so they look like a single object. They don't look like an X-shaped thing, because they just haven't gotten to that point in the cell cycle yet. So those are 4 double helices, a pair and a pair, the pair from dad and the pair from mom, and the same on the right side a little bit later in the cell cycle. So we'll continue this conversation and move on when we get to next class. Camera lucida, I'm going to draw what I thought I circled there. So I circled something that looked like that. Okay, 2 vertical stick-shaped things. Those are metaphase chromosomes. And I know that, and you know that, because it's only at metaphase that we can see chromosomes condensed, like individual objects. I also circled, on the right side, something that looked like that. An X-shaped object. That's what we usually think about when we think about chromosomes and karyograms, and if you think about what a nucleus looks like, when you take pictures of chromosomes, it looks like that. That is also metaphase, because again, this is the only part of the cell cycle where you can observe chromosomes as distinct objects. The difference is exactly what time in metaphase we're looking. The top is early, the bottom is later. So the previous pictures were 2 human cells, nuclei, chromosomes of 2 human cells, at slightly different stages of metaphase. And that makes all the difference. And again, this can be confusing, which is why I spend so much time focusing on this in particular. The object on the bottom, if I pulled this apart like I was anaphase, part of the cell cycle where the 2 homologs separate and get pulled into daughter cells, one side of the X is one double helix. The other side of the X is another double helix in horrible fat finger method. So the X-shaped thing is two double helices. It's two chromosomes side by side. It's two double helices side by side. You might have thought, which could also be true, you might have thought that it actually is two chromosomes crossing each other at the middle, from upper left to lower right, and from top right to lower left. That is also possibly true. But it's two double helices next to each other. But at this point in the cell cycle, late metaphase, what happens right after metaphase? Anaphase is when
They split up.
the cells start to divide, the chromosomes start being pulled into the two daughter cells, like I diagrammed there with the black arrows. These two chromosomes are starting to be separated. And at this stage in the cell cycle, those two chromosomes are only held physically together at the centromere. That's why it looks like the arms of the chromosomes, the tops and the bottoms, are spread apart, because the only physical attachment left between those two double helices is at the centromere. And that's how we can tell where the centromere is in metaphase. We can look at the shape of the chromosome and see where the arms are not separated. We talked about that earlier. Early in metaphase, what happens is there are actually two double helices lying right next to each other, so closely that you can't actually tell they're two double helices next to each other, because they're connected with proteins all down in between the two pairs of double helices. That is, they don't spread, the arms don't spread apart because the chromosomes are physically, the two double helices are physically glued together by this red protein. It's called cohesin, because it's cohesive, it's what keeps them bound together. That's early metaphase. So what happens from early metaphase to late metaphase is those red proteins, the cohesins, get removed, in this case, from the top part and from the bottom part. And that leaves only the little bit of cohesin that's at the centromere left. Put it another way. If we zoom into what we thought was a single stick-shaped thing in the previous slide, it's actually two double helices. Each of those things that look like an individual unit is two double helices. But they're lying so closely together, they're synapsed, they're stuck to each other, so that they look like a single thing. So let's go back to the previous picture, ask some questions if you want. And we'll finish up by counting the double helices and making sure we all agree on how many double helices. Please pay attention to biology class, but it's important to remember the cell cycle. Every cell has a couple of growth phases, and then there's synthesis phase, when chromosomes replicate. And then there's mitosis. That's cell division. There's a G1, G2 growth phase in there as well, but that's not as important as the fact that DNA replicates and then the cell divides. And we're going to be doing an exercise on this next week, maybe Friday. So we'll look at this a little bit more. But there's one particular part of mitosis, the stages of mitosis: prophase, and then metaphase, and then anaphase, and then telophase. You don't need to remember that. That is the stages of mitosis. And all mitosis is about what's happening to the chromosomes when the cell is preparing to divide. Metaphase is the part in the cell cycle where the chromosomes condense. The nucleus packs the chromosomes into little nice bundles like what we saw in the previous slide, so that when they get dragged into daughter cells, they don't break or tangle up with each other. That's the only time during the cell cycle that you will see distinct chromosome-shaped objects. To what to do for next time. We'll watch a couple more videos, try a couple exercises, and then in a week, by the end of the weekend, there's another discussion board item to do.